TIDE, or Tumor Immune Dysfunction and Exclusion, is a computational framework developed by the Shirley Liu Lab. The query gene function provides data-driven gene signatures of T-cell dysfunction and immunotherapy resistance through computations from a large amount of cancer genomics datasets. In this video tutorial, you will learn how to use the query gene function on the TIDE website. First, type a gene name such as PDL1 into the search bar and click the blue search button. You will then be redirected to a page where you can select the specific gene name that you're trying to search for. Many times this is not applicable, but for some genes with multiple aliases such as PD1, you will need to select a more specific gene name at this page. After clicking Continue, you will arrive at a page with several tabs. We listed the high confidence results in the core tab based on our core datasets in the T-cell dysfunction analysis. We also listed results computed from all collected datasets that profiled the gene expression, copy number, DNA methylation on gene promoters, protein level, and somatic mutations if applicable. Although this additional data may not be of high quality compared to the core sets, they may still reveal valuable insights of T-cell dysfunction in a specific cancer type. We also included several reference results, including REF therapy and REF screen, to show the gene-level association scores in immunotherapy clinical response and CRISPR screen of anti-cancer immunity. Please click each tab for an individual section. You can hover your mouse over any of the tabs or column titles for a more detailed description of the page or column. Also, if you would like to sort a column, you can just click on the title of the column you would like to sort. Lastly, if you see an eye icon in any cell of a table, you can click the icon to open a graphic survival plot. If you click the eye icon next to the gene name that you entered at the top of the page, it will bring you to a page on the GeneCards database with details about the gene. Now I will do a more detailed overview on the pages that correspond to each tab in their respective tables. The first page, Core, shows the most confident results using gene expression data. The first column on this table simply lists the data cohorts. Next, the cancer column lists the type of cancer of each data cohort, and the subtype column lists the cancer subtypes if applicable. The CTL correlation column lists the Pearson correlation between the gene level and the infiltration level of cytotoxic T lymphocytes. The T-cell dysfunction score is the z-score of the interaction effect between CTL level and gene status in the Cox proportional hazards or Cox-PH model. A higher score indicates that a cancer sample with high expression level of the query gene tends to be enriched in T-cell dysfunction phenotype. In contrast, a lower score indicates that a sample with low expression value of the gene tends to be enriched in T-cell functional phenotype. The risk column lists the survival risk score, which is the z-score for the gene effect on death risk in the Cox-PH model. A higher risk score indicates the gene activation to be associated with high death risk, thus short survival duration. The adjusted risk column lists the survival risk score that has been adjusted for the effect of cytotoxic T lymphocyte. Many genes are highly associated with death risk because these genes have high correlations with the cytotoxic T lymphocyte infiltration. Therefore, we computed a risk z-score with the cytotoxic T lymphocyte level as one background factor in the Cox-PH regression. Lastly, the count column lists the sample count in each dataset. In addition to the data cohorts that are currently available here, we will continue to add studies not only for this query gene model, but for the prioritized gene set and biomarker evaluation models as well. The expression page displays all results using gene expression data that we collected from TCGA, Metabric, Precog, and NCBI Geo databases. The survival column on this table lists the survival endpoint type of each data cohort, where OS means overall survival and PFS means progression-free survival. The next page, copy number, displays all results using copy number alteration data. Next, the methylation tab shows all results using promoter DNA methylation data. Since the columns for the copy number and methylation pages are identical to the ones in the core page, please refer back to that portion of the video for information regarding those columns. The exclusion tab displays the T-cell exclusion signatures, which are gene expression profiles from immunosuppressive cell types. The first column on this table lists the experimental conditions, while the second lists the z-scores computed as expression values normalized by mean and standard deviations in the same expression profile. Next, the reference therapy page creates a table that shows the association between gene expression and therapy outcome in clinical studies of immune checkpoint blockade. For more information about these columns, refer back to the description of the columns on the core page. Lastly, the reference screen page shows the gene knockout phenotype from genetic screens profiling regulators of T-cell mediated tumor killing or immunotherapy resistance. The condition column lists the experimental condition and the model column lists the cell models. The log2 fold change represents the difference of CRISPR guide RNA frequency between immune active conditions and control conditions. For each gene, we only show the median value among several guides. Since the log FC value distributions are very different among studies, we also normalize these values into z-scores through the mean and standard deviation of log FC within each CRISPR screen profile. You now know how to use the query gene function on the TIDE website. For more information, you can read the TIDE paper that is linked down below or visit the FAQ page on the TIDE website. Please also feel free to leave any questions in the comments below. Thanks for watching.